Hello, I am Sophia, and today we're going to talk about photosynthesis. So look at this image over here. What is here? What's important? Well, here you see some bugs, and you also see a leaf. So those bugs, they benefit by eating the leaf. And now, let me work on a slideshow about photosynthesis. Photosynthesis. So here we have some examples of photosynthesis. For example, the first image that we have here, this is a fern, a plant. The second image is kelp, which is an algae. The third image is a bacteria. Also, the fourth image is this image, the cyanobacteria. And the fifth image is a purple sulfur bacteria. Here we see the location of photosynthesis. So first we see this is the leaf, and here we have the upper epidermis of the leaf, the mesophyll of the leaf, and the lower epidermis of the leaf, where the CO2 comes in, and the O2 comes out. Here you see we've gone into one mesophyll cell. Here they contain nucleus, ribosomes, mitochondria, and vacuole, and we get into a chloroplast. Here's the chloroplast itself, and here's an area next to the thylakoid membrane containing a little bit of stroma. Okay, what is the equation for photosynthesis? So now let me first go to another slideshow that will cover a lot of this material. First, the equation for photosynthesis producing glucose is 6CO2 12H2O plus the light energy coming in yields C6H12O6 or glucose plus 6O2 plus 6H2O. Van Nielsen determined the general equation for photosynthesis different through examining the general equations for photosynthesis in plants and sulfur bacteria. In sulfur bacteria, we have an equation, CO2 plus 2H2S yields CH2O, which is the general formula for a carbohydrate, plus H2O plus 2 sulfurs. The equation for photosynthesis in plants is CO2 plus 2H2O yields CH2O plus H2O plus O2. The general equation for photosynthesis for all organisms that contain a chloroplast that looks like this is CO2 plus 2H2X yields CH2O plus H2O plus 2X. Plants split H2O as a source of electrons from hydrogen atoms and release O2 as a byproduct. Here are the equations. And here's just a confirmation that the atoms come here and they track the atoms through photosynthesis. CO2 becomes reduced to C6H12O6 and H2O becomes oxidized to O2, like you see in this diagram. Here, this is just a video that you could watch which demonstrates the whole process of photosynthesis. But let's go to the next page. Here there's a summary of how both processes partner. Now we need to get back into the slideshow. Here there's the image of how light reactions in Calvin cycle partner. Here there's the light reactions, and here there's a Calvin cycle. Here they say H2O is an input, oxygen is an output. It produces ATP and NADPH from light energy. Leaves are green because they absorb blue and red light and reflect green light. There's an image of the electromagnetic spectrum on the next page of the slideshow. Here you see an image of the electromagnetic spectrum of the wavelength. Going back, And there's also an image in the Campbell Biology textbook, 11th edition, of what the compound chlorophyll A absorbs. It absorbs blue and red and reflects green, as you see in this image. Now let me go back to the other slideshow. When a molecule absorbs a photon of light, one of chlorophyll's electrons gets excited to a higher energy state. Or there's a misspelling, there should be a C after the X. And then there's a process of electron excitation. Now we're ahead. Now we're here in the slideshow. Here you see the photon and the chlorophyll molecule get excited and release heat and photons. This is an image from Campbell Biology. Once absorption of a photon raises the electron to an excited state, the electron cannot stay there for long. As excited electrons fall back to the ground state, photons are given off, an afterglow called fluorescence. The process of fluorescence is demonstrated in the next image of the slide, but it's quite poor quality. So I'll show you the process of fluorescence here. See, this is an image through ultraviolet light. This is the process of fluorescence, see? And within a billionth of a second it releases item, and now, see, it's now fluorescent. Okay, now, what comes next? There's part of the light-dependent reactions in one photosystem in my slideshow. Here you see the second image is the important image here. We have light that passes in, and the stroma, and the photosystem, and the primary electron acceptor. Here's the chlorophyll molecule, and the reaction center that takes an H2O and divides it into half O2 and 2 hydrogens. Here's the thylakoid membrane. It looks like the phospholipid bilayer, but it isn't. It's a different membrane. Now see this image over here. Here we see light, photosystem 2, the electron transport chain, and then photosystem 1. Here it passes through a molecule called plastocyanin, and here it passes through ferredoxin. Here we have an enzyme called NADPH reductase. It isn't important to remember the name. Here you have the ADP gets turned to ATP via ATP synthase. Now we're ahead in this slideshow. Cyclic electron flow does not produce NADPH and does not release oxygen. On the other hand, it generates ATP. See the process of cyclic electron flow. Here you see the process of cyclic electron flow here. We have these dots and the cytochrome complex. It passes through the ferredoxin and plastocyanin and it only uses photosystem 1. It does not generate NADPH because it didn't get here, and it also doesn't include photosystem 2, so it doesn't generate any additional water. The location of the thylakoid membrane's components are not linear. The real location of the thylakoid membrane is different. You see the real image containing the real locations of the thylakoid membrane, here and here. But unfortunately, it's so poor quality. They'll show you this in the actual book. We're already ahead. See here, these are the real locations in the thylakoid membrane. Now here you see the thylakoid membrane and the stroma, and where these are located inside the chloroplast. Now we are going back to the Calvin cycle. This is almost the last image in the whole slideshow. So what is the Calvin cycle? We see this is the Calvin cycle. We have 3 molecules of CO2, and there's carbon fixation, and there's an enzyme called rubisco that generates a molecule called RUBP. And then there's stage 2, which is reduction. They consume 6 ATPs and 6 NADPHs. It generates 1 G3P, or a 3-carbon sugar. It regenerates RUBP, spending ATPs. 5 molecules of RUBP come back. 6 molecules of RUBP are generated. And now we'll talk about photorespiration, because this is important. There's another alternative cycle that uses oxygen that spends of ATP and does not create the G3P. Because there are 5 molecules, and 5 molecules of RUBP are supposed to be regenerated. This cycle is important because what we see is that it doesn't generate anything. If it were not to be a cycle, it would generate more G3Ps than the Calvin cycle, and it would be an alternative, more efficient pathway. But it is a cycle. The RUBP is supposed to be regenerated. The RUBP is not very common. There are alternative ways to deal with photorespiration. For example, here we have a 4-carbon called oxaloacetate. How does it make that? It uses CO2 that comes into PEP carboxylase. And then here oxaloacetate, and then it produces malate. Malate gets into the bundle cells, and then it conducts a regular Calvin cycle on the previous slide. Then it gets consumed as a vascular tissue. And also, ATPs get spent in this process. But then what happens is that they also produce ATPs here. How? There's only photosystem 1 in these bundle cells that don't have access to either O2 or CO2. But what gets transferred is pyruvate. And also, the process with PEP carboxylase releases PEP. What happens is that we reproduce the oxaloacetate, and this new cycle is a cycle. Now let's look at CAM photosynthesis. Here we have almost the whole chapter. We have CO2 that comes through an organic acid and gets placed in vascular tissues. Now the CO2 gets used as a Calvin cycle, and it produces sugar during the day. So now they use alternative times. Now let's look at the cycle of photosynthesis. The light reactions are carried out by molecules in the thylakoid membranes. They convert light energy to the chemical energy of ATP and NADPH. They split H2O and release O2 to the atmosphere. This saves us from the oxygen-carbon dioxide problem, the global warming problem. So if the plants won't be cut, then the global warming problem would be less severe. In the Calvin cycle, it takes place in the stroma. It uses ATP and NADPH to convert CO2 to the sugar, G3P. It returns ADP, inorganic phosphate, and NADP to the light reactions. So both the light reactions and the Calvin cycle are dependent. Thank you. Bye.